Dear students, Assalamu alaikum. Hoping you all will be fine. Today we will discuss about the building blocks of the protein. Amino acids have a common feature that permit them to be linked together into the polypeptide chains in uncommon characteristics that give each polypeptide chain its unique feature. 20 commonly known amino acids, which are the alpha amino acids, are present in the formation of the protein. Which are required. We can say there are 20 amino acids which are required to make the proteins. The unusual amino acids also found in the protein results from the modification of the common amino acids. In a few cases, these amino acids are incorporated directly into the polypeptide chains during the synthesis. Most frequently, the amino acid is modified after incorporation. The unusual amino acids found in non-protein components are extremely varied. At the central tetrahedral carbon atom called the alpha carbon. The unusual amino acids found in the non-protein components are extremely varied in type and are formed by a number of different metabolic pathways. The amino acid contain a central carbon atom, which is actually the tetrahedral carbon atom, and this tetrahedral carbon atom is known as the alpha carbon. Amino acids, the name of the amino acid is commonly derived from the source where they are isolated. Say, for example, asparagine is derived from the asparagus, first discovered in 1806, last discovered in amino acid is threonine in 1938. Glutamate from wheat, gluten, tyrosine from the tyrose, Greek cheese, and glycine from the sweet glycose, Greek. These are the Greek words. These are the sources from where they are isolated. All 20 amino acids are actually the alpha amino acids. Have carboxyl group and amino group bonded to the same carbon atoms called the alpha carbon. Amino acids differ on the basis of the side chain, which is known as the alkyl chain. Our side chains vary on the basis of the size, structure, and the electric charge on them. There are three features which differentiate the amino acids from one another. Um, Commonly, amino acids are assigned three lettered abbreviation and one letter symbol. Say, for example, glycine. For example, glycine contains the three letter symbols. Say, for example, gly, three letter abbreviation. This is the abbreviation, and its symbol is G. Say, for example, so this type of symbol are assigned to the amino acids present in nature. Proteins are present in the diet, or they are required to digest. They are required to they are required for the proper functioning of the body. They are present in the enzymes. And there are some hormones which are made up of the protein. All the proteins are made up of the amino acids. The carbon, which is central of the chiral carbon, all the amino acids except the glycine are the alpha carbon bonded to the four different groups. All the amino acids are alpha amino acids, or you can say all the amino acids are contain the chiral center except the glycine. I already told you that the chiral carbon is the carbon which is bonded with the four different atoms. Carboxyl group, they contain, all the amino acids contain carboxyl group, amino group, alkyl chain, and the hydrogen atom. Four different atoms occupy two unique arrangements, two possible stereoisomers, mirror images like the D isomer and the L isomer. Mostly L form exist. D amino acids are in small peptides, maybe in the bacterial cell wall and the peptide antibodies. Cells specifically synthesize uh, cells specifically synthesize L isomer because active sites of the enzymes are asymmetric. Amino acid is composed on, for example, amino acid is composed on central carbon atoms, which is central carbon atom. Amino acids are composed of central carbon atom. This is central carbon atom. On one side, it contains the hydrogen alkyl group and amine group and the carboxyl group. Sometimes the amide group contains the positive charge, and if this hydrogen is removed, the carboxyl group will contain the negative charge. So sometimes it can act as a base and it can act as an acid. We will discuss about this later. Amino acid can be classified into the five major groups on the basis of the properties of the alkyl group, on the basis of the polarity, and to react with the water at the biological pH. The first group is the non-aliphatic amino acids. In this group, around seven amino acids are present: glycine, alanine, proline, valine, leucine, isoleucine, and the methionine. This group contains the non-polar and the hydrophobic alkyl group. This group tend to cluster together because these are the hydrophobic groups. The property of the hydrophobic group is to cluster or to point together. They make the cluster. Stabilized structure of the protein. They are involved in stabilizing it's the structure of the protein by hydrophobic interactions. The examples are alanine, valine, leucine, isoleucine, glycine, isoleucine, etc. Among these amino acids, glycine is the simplest amino acid. It is a simple structure. Methionine is the sulfur-containing amino acid. It contains the thioether group. If you look on the methionine here, the methionine contains the thioether group. In the side chains, proline is the aliphatic. Proline is the aliphatic. Can contain the aliphatic side chain. It contains a ring-type structure, and it also contains a secondary amino group, secondary amino or amino group, as you can see. Secondary amino or amino group it contains, which is the part of the which is part of the ring. Second one are the aromatic alkyl chains containing amino acids. Among these, three amino acids are present: phenylalanine, tyrosine, and the tryptophan. These are relatively non-polar hydrophobic amino acid side chains. It can interact in the hydrophobic interactions. Hydroxyl group of the tyrosine can participate in the hydroxyl group. This is hydroxyl group can participate in the hydrogen bonding. This is important function in the some enzymes. This is sometimes it makes the catalytic site of the enzymes. Tyrosine and the tryptophan are the more polar than the phenylalanine. Because of the hydroxyl group of the tyrosine, because uh, tyrosine contains the hydroxyl group and it is containing the nitrogen. So these are the more polar as compared to the phenylalanine. Tryptophan, tyrosine, and phenylalanine, these are all absorb the light at 280 nanometer wavelength. This property is used for the characterization of the protein. Aromatic amino acids absorb light in the nuclear ultraviolet range. The aromatic amino acids phenylalanine, tyrosine, and tryptophan are possible absorption maxima in the near ultraviolet. The absorption band arises from the interaction of radiation with the electrons in the aromatic ring. So on the basis of this feature, we can on the basis of this feature, we can measure the concentration of the protein. The near UV absorption properties of the proteins are determined solely by their content of the three aromatic amino acids. Next are the polar uncharged R group containing amino acid. There are five amino acids: serine, threonine, cysteine, asparagine, and the glutamine. These are the polar uncharged. These are the polar molecule, but these are the uncharged molecule. Because of the equality of the charge, they cannot show the charge in the they cannot have the overall charge. The charge are because of the charge R group are soluble in water. These are the hydrophilic compounds. Functional group make H bonding with water. Serine and threonine polarity is contributed by their hydroxyl group. If you look on this, serine has the hydroxyl group and threonine has the hydroxyl group. The polarity is contributed by this one. Asparagine and the glutamine. Asparagine and the glutamine. Amide groups are present in both of these. As you can see, the amide groups are present. And the cysteine contains the sulfur thiol group. It is containing. So the polarity is because of the um, this can take part in the polarity. Cysteine is the outlier polarity by the sulfhydryl group. It is containing the polarity because of its sulfhydryl group. This is important to make the disulfide linkage. As you can see, this is the cysteine molecule. If the two cysteine molecules, if the two cysteine residues come close to each other, they release their hydrogen and make a covalent link between between each other. So this link is known as the disulfide bridge. The next two are the positively charged alkyl groups and the negatively charged alkyl groups. Among the positively charged alkyl groups, lysine, arginine, and the histidine are present, and in the negative molecules, negatively charged amino acids, aspartate, and the glutamate are present. The negative charge of these two groups is because of the extra carboxyl group present in these molecules. Extra carboxyl group, because of this, these extra carboxyl groups are present, so they contain the negative charge. The positively charged molecules, the positively charged residues, lysine, arginine, and the histidine, they contain a significant positive charge at the pH 7. Lysine has a second amine at at epsilon position in the aliphatic chain. Second amine group lysine, as you see, lysine has a positive charge because of its extra amine group. Arginine has a positively charged guanidinium group. Positively charged arginine contain the positively charged guanidinium group. This one, as you can see here, because of this one, this is it contains the positive charge. And histidine has a aromatic amidazole group. This one, amidazole group. So because of this, there is a positive charge present on these molecules. We can also classify the amino acid into three major classes, but this is normal: hydrophobic, hydrophilic, and the special amino acids. One with reacting with water, other which are not reacting with water, and the other ones are the special with the cysteine containing the sulfur group, glycine containing no chiral carbon, and protein being an aliphatic containing butane are the amino group. Tomorrow we will discuss about the acid and the base properties of the amino acids. Good luck.